Hello students, welcome back to my channel CSIR Buddy. Here we are with another video of Developmental Biology Part 2. So these are the topics for upcoming videos which we are going to study. Today we will deal with commitment, specification and determination. Moving forward, what is cell fate? So very first topic is cell fate, then we are going to study about the commitment, then other types of specification and all. So coming back to our topic, cell fate. What is fate? सेल आगे चलकर क्या बनेगा? Like we have a stem cell. स्टेम सेल आगे चलकर किस सेल में कन्वर्ट होगा? दैट इज नोन एज सेल फेट. सो सेल फेट इज द प्री डिटर्मिनेशन ऑफ अ सेल टाइप. सो इफ अ सेल, stem cell is determined to become a muscle cell, then this is known as its fate. That is, muscle cell is its fate. The fate of a cell is defined as all the different cell types its descendants can become during a normal development. Cell fate becomes more restricted until a cell is terminally differentiated. जब तक सेल डिफ्रेंशिएट हो जाता है, टर्मिनली डिफ्रेंशिएटेड हो जाता है, तब उसकी जो फेट है वो और भी रिस्ट्रिक्टेड हो जाती है. इफ़ अ सेल फेट इज़ टू बिकम अ नर्व सेल, देन coming towards the differentiated state, it becomes more restricted, that now the cell is going to become a nerve cell only. So this was all about the cell fate. Then what is commitment? Commitment decides the fate of cell to specific induction and their capacity to develop into a particular kind of cell. Commitment, जब एक सेल कमिट करता है, like एक स्टेम सेल है and it has committed now to become a brain cell, then this is known as commitment. Stages of commitment. So there are two stages of commitment. One is specification, another is determination. So what is specification? First, the cell gets specified to become a certain kind of cell. Say for example a nerve cell. But specification, this particular stage is a labile stage. Labile stage means it's not permanent. That means the cell could become another type of cell, or it can change its fate to some other cell. Say a cell was committed to become a nerve cell during its specification stage, but now it has changed his fate because it was not determined, so it can change into muscle cell. Now what is determination? This one is a permanent stage where the cell is now determined, and now if a cell was determined to become a muscle cell, then it has no going back and it will only become a muscle cell only. So this was the basic difference between specification and determination. So specification is specified, it is labile, not permanent, but determination, now cell is determined to become a particular cell type. So when we take a zygote stage get due to mitosis get multiplied and gets converted into an embryo. Now embryo has the potency to become so many types of cell. Like this property is known as totipotent. Potency we have already studied in the last video. If you haven't watched that video, so first watch that video, then only you will get to know the meaning of this word. So totipotent means now these cells could be converted into so many cell types. Maybe a cell can become a nerve cell, some other may become a heart cell, or whatever. So these specification and determination, these all are known as cell fate. So I think this topic is clear till now. Moving further. So this is a diagrammatic explanation. We have an undifferentiated cell. Undifferentiated cell is going towards commitment. And commitment, we have already seen two types of commitment. First, the cell gets specified. So this is specification stage. And I have mentioned that this stage is labile, that is not permanent. Now this stage is determination, that now the cell is determined and there is no going back. So once a cell gets determined, then it cannot change its fate. So this is the undifferentiated cell. Now the cell has been specified into a brain cell. So this stage is specification stage, which is labile, not permanent. Then the brain cell is now determined due to induction, and this inductive signal could be of any kind, like morphogen signal or any other kind of signal. So now the cell is going to be determined. So determined, like brain cell, first it was specified, now it is determined. Like from here there is no going back. So the cell will obviously convert into 100% brain cell only. So this stage, when a determined cell gets converted into brain cell and there is no going back, then the cell is known to be differentiated. Now it has been differentiated. So I think this concept is very much clear and this is very important. Now, types of specification. There are main three types of specification. First, autonomous specification, conditional specification, and syncytial specification. So the first one is autonomous specification. Cell which do not change their fate according to their surrounding. So they are presumptively specified, like they have some cytoplasmic determinants which are already present in the cell, which are determining their fate. So the fate is predetermined and they do not change their fate because of their surrounding. This shows a mosaic type of development. Again, very important, it has been asked. Prospective potency here is equal to its prospective fate. So potency is equal to its prospective fate, like there is no change. जो उसकी सेल की पोटेंसी थी, जिस टाइप का सेल बनने का, तो वही उसका फेट रहेगा. Nothing can change its fate. No environmental condition would be able to change its fate. So there is no influence of any environmental condition over here. It is seen in invertebrates, or we can say in lower organisms. Now coming towards the example. If we took a muscle cell which was autonomously specified, so and we put this into a different environment, like we are putting this into a different environment, that is a liver cell, then also it will not change its fate due to its surrounding and become a muscle cell only. It won't change its fate because of its surrounding. So this is known as autonomous specification. We also have seen the example. So it is, I think it's clear to you. Conditional specification. The name suggests itself. The name is suggesting that conditional. Conditional means any external condition would affect its main fate. Cell is not determined over here. Prospective potency is higher than the prospective fate. So potency is higher than fate. So the cell may change its potency. Earlier it was becoming a brain cell. Now if we put it into another situation, another condition, it may change into a nerve cell. So environmental condition or the condition of the surrounding is affecting that. So what will happen? Again, an example. If we take a brain cell which is a brain cell, and we put that cell into a liver cell, then it will change. Earlier it was conditioned to become brain cell, but now if we put it into a different environment, then it will change its prospective fate and become a liver cell. So from here we get this thing that potency is higher than its fate. Cell change their fate in accordance with the surrounding. Found in all vertebrates. This is very important. All vertebrates, that is higher organism. Morphogen gradient is involved. Now what is morphogen? We will be studying in our upcoming videos in detail what is morphogen. For now, morphogen is a type of signal which can give a signal to a cell to convert into a particular cell type. So conditional specification में क्या होता है कि या तो cell to cell interaction होगा in a juxtacrine manner, that is juxtacrine signaling. Juxtacrine signaling is that signaling in which cell to cell जब interaction होता है then they pass on signals. So conditional specification can be done due to this type of juxtacrine signaling. Or we have seen morphogen gradient due to morphogen gradient also, or any external environmental effect also. It can change its prospective fate. Now moving further to syncytial specification. Syncytial specification, it is an example of Drosophila. Its example is Drosophila. So in Drosophila what happens is there is only nuclear division. Only nuclei are dividing. That means the karyokinases, which is known as nuclear division is not followed by any cytokinases, that is cell division. This cell undergoes cell division but after a very long period. After several karyokinesis, then we can see a cell division. So nuclear division without cell division result in cytoplasm with many nuclei, because it is only undergoing karyokinesis and karyokinesis is not followed by cytokinesis. Now in this case, in the early stage it is showing conditional specification, but in the later stages it gets autonomously specified. So Drosophila development, when we will study about the Drosophila development, then we will talk about this topic in detail in our upcoming video. So guys, if you like this video, then subscribe to our channel, share this video with your friends and press the like button.